This project is studying the role of O-GlcNAcylation in pituitary adenomas. These tumors are derived from the endocrine tissue of the pituitary gland, which, as you can see at the bottom left, sits at the base of the brain in a tight bit of anatomy. Because of this tight anatomical space, these tumors can quickly exert a mass effect, which results in profound neurologic dysfunction. Additionally, due to the unregulated secretion of pituitary hormones by these tumors, patients can experience profound endocrine disruptions as well. Recent studies in genetic and epigenetic factors have been unrevealing regarding the etiology of the tumorigenesis and hypersecretory function of these tumors. We've therefore hypothesized that a more dynamic change in the cells, such as shifts in O-GlcNAcylation, may be responsible for the dysregulation of these cells. At right, we can review the normal physiology of these cells, whereby CRH via a GPCR activates a kinase cascade, resulting in the transcription of POMC mRNA being translated to POMC protein and eventually cleaved to release the ACTH peptide hormone secreted from the pituitary gland. We started by measuring the levels of OGT and OGA expression in human pituitary adenomas. OGT and OGA, of course, are the enzymes responsible for the addition and removal of O-GlcNAc. We found that compared to control pituitary tissue, both the non-secreting subtype of pituitary adenoma and in particular the ACTH secreting type or corticotropic pituitary adenomas had an elevated expression of both of these enzymes responsible for O-GlcNAc cycling. In particular, one tumor which was very clinically aggressive, seen at right, had even more exaggerated expression of OGT and OGA, suggesting to us that increased O-GlcNAc cycling is an integral component of tumor function and growth in human pituitary adenomas. We then transitioned into a mirroring cell model to study how CRH induces POMC expression and where in this pathway O-GlcNAc might modify this signaling. We found on the left that in standard glucose conditions, whereby CRH typically results in POMC expression, inhibiting OGT and therefore decreasing O-GlcNAcylation resulted in a loss of responsiveness to CRH. In low glucose conditions, on the other hand, where there is no baseline CRH responsiveness, we found that we could recover CRH responsiveness by treating with OGA inhibitors, increasing the level of O-GlcNAcylation within these cells. To determine whether or not this regulatory step was at the level of POMC promoter activation, we repeated this study using a plasmid and dual luciferase activation assay. However, when we inhibited OGT, we did not prevent CRH responsiveness, and when we increased O-GlcNAcylation in low glucose conditions, we did not recover CRH responsiveness, indicating that this level of regulation is post-transcriptional. We therefore hypothesized that the level at which POMC mRNA expression is increased by CRH is at the level of mRNA stabilization. Indeed, in standard glucose conditions, we do see that cells where new transcription is inhibited still stabilize POMC mRNA in response to CRH. However, when these same cells are co-treated with CRH and OSMI4, we see a loss in the stabilizing effect as seen by the red dotted line. In low glucose conditions, where POMC mRNA is very unstable, we see that increasing O-GlcNAcylation using thiamine G to inhibit OGA does indeed increase POMC mRNA stability. In order to better understand which pathways were involved in the O-GlcNAc dependent stabilization of POMC mRNA, we used mass spectrometry of cells treated with CRH and untreated cells to see which pathways were more or less O-GlcNAcylated in the context of stimulation by CRH. We found particularly that pathways involved in mRNA splicing were enriched with O-GlcNAcylation, and pathways involved with nonsense-mediated decay were depleted in CRH-treated cells. This suggested to us that the stabilization is occurring through the increased proper splicing of POMC mRNA and associated decreased decay of incorrectly spliced mRNA.